Now we're going to talk about branched chain amino acids. So these tend to be in our supplements, used as supplements because they do help you build strong muscles, and that's why you see usually some muscular, somebody's trying to sell these. So they are essential. You do need to get all your branched chain amino acids in your diet, and they are metabolized by the liver or by the muscles rather than the liver. So you don't really need supplements. You just need a really good diet. But here they are. Isoleucine, leucine, and valine are your branched chain amino acids. Majority of the metabolism occurs in the muscles, and that's because the aminotransferases are found in the muscles. Not so much in the liver, but definitely in the muscle. And you need these aminotransferases to make the alpha keto acids. So the first step is going to be the transamination of the branched chain amino acids in order to start breaking them down. Breaking down these branched chain amino acids requires the transamination enzymes. And then you have to get rid of, when you're fully breaking them down, you have to do oxidative deamination and to get rid of that amine group. And so this, you need the alpha branched chain amino acid alpha keto acid dehydrogenase, BKCD. So BCKD stands for the branched chain amino acid keto acid dehydrogenase. So the BC just stands for the branched chain amino acids. K stands for the keto acid, and then the dehydrogenase. To make these are to make the metabolites of branched chain amino acids. And the reason I mention this enzyme, BCKD, is because if you lack this enzyme or have a genetic defect in this enzyme, then that can result in the maple syrup disease. So maple syrup disease is where you get the urine has the sweet odor to it, a maple syrup smell to it. And that's caused by the accumulation of these alpha keto acids. So when the muscles are trying to break down these branched chain amino acids, if they do not have this enzyme or there's a genetic defect in this dehydrogenase, this BCKD, this dehydrogenase, which is going to fully break down the branched chain amino acids, if you don't have that, then the metabolites of branched chain amino acids will accumulate. You can't fully remove them from the body. And we're going to talk about that next week, or not next week, in the next lecture, actually. It's also in that one. So there's going to be a little overlap in the next lecture, too. But that's going to cause maple syrup disease and cause issues with the muscles. So we'll talk about that in a minute. Here's the degradation pathway of your branched chain amino acids. Again, essentially the first step is transamination. That's where you're going to start making the keto acids. And then you have to get rid of the amine group, that oxidative decarboxylation, to fully get rid of the amine group. But this is where the problem comes in because you don't have the dehydrogenases. Branched chain, oxidative decarboxylate, the branched chain amino acids. Essentially, you cannot do this step. You can't break them down, and so you get a buildup of these alpha keto acids from these branched chain amino acids. And that's what's going to cause issues in the body, ultimately leading to maple syrup disease. So it's these guys. So these guys will accumulate, the branched chain amino acids, leucine, valine, and isoleucine, and their metabolites, these alpha keto acids, will accumulate. So here it is. It's not in the next lecture. Maple syrup disease, so it's a rare disease. It is, but this is probably one you might see on the boards. Autosomal recessive disorder, so it's related to these branched chain amino acids. It's accumulation of the alpha keto acids. So you can do the transamination step, but then you can't get rid of the alpha keto acid because that's what you need the BCKD, the branched chain keto acid dehydrogenase. To get rid of the keto acids, you need that enzyme. And if that enzyme is defective, then it's going to result in maple syrup disease, essentially feeding problems, vomiting, ketoacidosis, muscle and neurological problems, and eventually the reason why it's called maple syrup disease is because one of one of the branched chain amino acids, isoleucine, will accumulate in the urine, and it smells like maple syrup, apparently. Never smelled it before. But it's related again to the fact that you can't get rid of these alpha keto acids because you're missing that dehydrogenase to get rid of the keto acids. Histidine. So histidine is also an important one. So we talked about a few of them now, but histidine is important for making, I believe this one's easy, histamine. Histamine, if hopefully you recall, maybe you've heard this before, histamine is related to allergies. So when you have that inflammatory allergic reaction, histamine is going to be released, and that's going to cause inflammation. That's going to cause the swelling, all the physiological pathways that are going to become, that are going to trigger inflammation. Infiltration of white blood cells, it's going to cause runny nose, watery eyes, it can cause the narrowing of the air passageways. So histamine is really important in the inflammation pathway. And because it's important in the inflammation pathway, it is what triggers allergies. And it's also a neurotransmitter in the brain, histamine is. But again, the main focus here, histidine can synthesize histamine, which is important for the inflammatory process, and also part of the issue related to allergy symptoms. And that's a lot of medicines try to target histamine release. So cystinuria is the most common one of these, but here's a few of these diseases related to amino acid metabolism. Some of these we talked about, actually we talked about all of them except for this one. Actually, we didn't talk about this one either. But don't have to memorize the slide. This is just showing you how common they are. That's not something I would ask you. Here's all these pathways, all these other diseases you can read about. Here's maple syrup disease, homocystinuria. I don't know where PKU is, but that's okay. And here's the chapter summary. This again, you don't have to read this, but it might be a nice summary for people if you want to read through it. It might be useful to you. I didn't add these chapter summaries to the slides. Another person did, but I kept them in here because I thought they might be useful. I haven't actually read them, so you'll have to take that with a grain of salt. And these pathways, I also did not add these pathways to the slides in case I hadn't told you guys yet. Again, it's just, depending on how your brain works, this might be useful to you when you're learning the material. But I did put these review slides in. So these are already here. What products are made from histidine, what is the branched chain keto dehydrogenase and its role in maple syrup disease, what is products made from cysteine. So again, if you go through this, so I start out, what kind of products are made from the degradation of amino acids? What does it mean for an amino acid to be essential, conventionally essential? What makes an amino acid glucogenic, ketogenic, or both? Which amino acids are ketogenic? So if you go through the review lecture videos and go through with these questions as you're walking through it, then that will really help you guys see what I'm trying to focus you on. And if not, you can ask me questions. All right, so we are going to go on to the next one, close without saving this one, because I'm going to pull out the other one, which is, I guess, number 7. Whatever that was. Amino acids and their special products. So we've already talked about a lot of these. So hopefully this last little portion will feel a little bit like review. So, because you already know, for example, what amino acid do we need to make melanin? Tyrosine. What amino acid do we need to make melatonin? Tryptophan. So again, we like a lot of things from amino acids. Some we've talked about, others we haven't. These are some of the things we are going to talk about, but not all of these, actually. We're going to talk about purines and pyrimidines, but not until later. We will talk about some hormones, some porphyrins, neurotransmitters, chapter is all about specialized products that we can make using amino acids. We're going to start with porphyrins. There's a collection of porphyrins. The only one I'm concerned with you guys remembering is heme. So porphyrins are up here. They're this picture here. So these are porphyrins. And notice what they look like. They have a lot of nitrogens. They're this really big ring structure with these nitrogens. And they usually have a mineral in the middle of them. So so they usually have a bind to a metal ion, and iron is a very common one. So for example, these often have an iron in the middle of it. The most prevalent heme or porphyrin is heme. So heme is found in hemoglobin, so that's why iron is a very common example. What is the role of iron in the middle of this heme? What does it do? It binds to what? Oxygen. So it binds to oxygen. So that's why this heme in hemoglobin and myoglobin as well, is really important because it binds to iron, which is going to bind to oxygen. So myoglobin and hemoglobin can carry and hold on to oxygen because of this heme. And heme is just an example of a porphyrin. It's the most prevalent one, the only one we're going to talk about. But of course, there is myoglobin. Cytochromes or porphyrins have this heme group unit to it. The cytochromes are really common. Well, they're common in the liver to get rid of various toxins and vitamins as well. You also have the cytochromes in the electron transport chain. So cytochromes are just compounds in the body used for a variety of things. But I'm going to focus you guys on heme, especially related to hemoglobin. That's really what we're going to focus on, heme related to hemoglobin. And heme is rapidly synthesized and degraded. This whole chapter is really long, talking about heme synthesis and degradation. I'm going to tell you exactly what you need to know, and not that much, not as much as the book wants you to know. So the synthesis of heme occurs in the liver and the bone marrow, and that's primarily because in the bone marrow, that's where hematopoiesis takes place. What is hematopoiesis? It is the production of what? Blood cells. White blood cells or red blood cells? Both. Okay, trick question. It was both. And those blood cells are going to, some of them are going to contain this heme component. So that's why the bone marrow does produce heme. It needs heme in order to make red blood cells, because red blood cells are full of hemoglobin. So hemoglobin, the carbons and nitrogen molecules are provided by glycine and succinyl-CoA. And I'll show you that in, I think, the next picture. No, this picture. So here you have glycine and succinyl-CoA are needed to provide the various components. Oh, so again, don't have to worry about inhibition or stimulation. We're not going to do those pathways in this lecture. But this is just showing you how glycine and succinyl are going to be providing the nitrogen and the carbons necessary for production of heme. I do not like the way this is set up. So I'm just going to move on to that. So here is a heme. You got all these nitrogens and of course all these carbons being provided. And this is porphyria. Porphyria is a genetic disease caused by abnormalities in enzymes involved in heme biosynthesis pathways. So you can imagine if we can't synthesize heme, that is going to cause issues, not only related to hemoglobin. So if we can't make enough hemoglobin, what do we end up with? Yeah. So what do we end up with if we can't make enough hemoglobin? Anemia. So anemia is going to be one issue, but also we need heme and porphyrins for other molecules as well. So you're going to have other impairments of the nervous system. So developmental delays, low IQ, neurological issues, anemia. It can be fatal, and this actually can be caused by lead poisoning. Lead can actually interfere with the enzymes needed to make heme. And so lead poisoning, not really a problem today. But it can be. Some people, we do still have instances of lead poisoning. It used to be a bigger problem before we realized lead was a poison. But nowadays we don't make as much stuff with lead, so we don't have as much issues with lead poisoning. But lead poisoning, the reason it's a poison is because it interferes with the enzymes that are making heme. And heme is required for a lot of stuff, not just hemoglobin. All right, this is what I want you to know about its degradation. So if you notice, I didn't really talk about anything about its synthesis. If you read the chapters, does anyone read the chapters? I don't blame you. Okay, so I don't have to worry about it. But yeah, this chapter, seriously, I took like tiny pieces of it and put it together. So if you do read or listen about this chapter, don't sweat all the details, because this is what I want you to know. So again, heme is a type of porphyrin, a really common one found in hemoglobin. It does have a lot of nitrogens and carbons. The nitrogens come from glycine, the carbons come from succinyl-CoA, and that's all I want you to know for its synthesis. There's a lot more to it, but that's what I want you to know. I do want you to also know that there is that disease state, porphyria, which is a genetic disorder related to an inability to make enough heme, because you don't have the enzymes are not working properly. And that can be a genetic disorder, but it can also be caused by lead poisoning. And then this is what I want you to know about its degradation. So heme, we're going to specifically relate it to hemoglobin, because that's where you're going to most be working or thinking about this when you're a medical practitioner. So your red blood cells are recycled. They every 120 days approximately. So that's a fast turnover for blood cells, for any cells. 120 days is a pretty fast turnover. So red blood cells are going to be degraded, and that is going to be by phagocytes, so like macrophages in the liver as well as the spleen. The liver and spleen, their function is to help recycle red blood cells. Mostly the spleen, but the liver also does it, especially if you lost your spleen, as some people do. It's a very fragile organ, and so when it is destroyed, the liver can take over the functions, most of the functions of the spleen. So that's where red blood cells are degraded, so you should know that it happens in the spleen and the liver. Heme is converted into bilirubin. Bilirubin is a red-orange color. If bilirubin increases in the body, it's going to cause I'm going to put it in a different color here. It's going to cause jaundice. And that's that yellow coloring of the body and the eyes. I always thought students automatically knew this, so I didn't teach it very strongly last term. But jaundice is caused by the inability to break down bilirubin. What is bilirubin? Bilirubin is a product of heme breakdown. So when you break down heme, you turn it into bilirubin. And bilirubin is sent to the liver, and that's going to be via albumin. Albumin doesn't just carry fatty acids, it carries other things too. It's a major transport protein in the blood, carries a variety of things. For example, bilirubin takes it to the liver for excretion via the bile. That's where we get rid of bilirubin. So heme, broken down into bilirubin, bilirubin travels to the liver via albumin, and once it gets to the liver, it's put into the bile, and we can excrete it. So excess is removed. If excess is not removed, we end up with jaundice. So I'll talk about that in a second here. This is essentially telling you the same kind of thing, or a few other things. I'm going to go to the next slide, and then I might come back to this. Yeah. So jaundice. So you can see the jaundice in the eyes here, the orange and yellow-orange of the eyes. That's a really apparent sign that someone has jaundice. Oftentimes, and jaundice is caused by a buildup of bilirubin, and that's a result of heme degradation. So bilirubin comes from heme degradation. It is based on some underlying disorder. So someone who has high levels of bilirubin probably has some sort of liver damage disease, or they could have obstruction of the bile duct. So if there's obstruction of the bile duct, you can't properly remove bilirubin via the bile or feces, and so the body is going to accumulate it. This is common-ish in newborns, so jaundice in newborns. And there's this little kid here over under the blue light. Essentially, the liver is going to, when the liver isn't working properly, it doesn't have all the enzymes ready yet. It can't conjugate bilirubin for bowel disposal because the liver is not making the enzyme yet, especially when the fetus is a preemie, born before the liver starts making the enzymes. Okay, back up. I'll say this better. So essentially, the liver has to conjugate bilirubin with something in order to put it to the bile. If the enzyme allowing the liver to conjugate bilirubin is not there, then you can't put bilirubin in the bile. And that's what happens with infants that are born that don't have this enzyme yet. They will have this enzyme, but the first few days, some infants don't have that enzyme working. And so they will have bilirubin build up because they can't properly conjugate it in order to put it into the bile. So what is this blue light? This fluorescent blue light is essentially there. That blue light affects the bilirubin in the body, causing it to become more water soluble naturally. And then that water soluble bilirubin can be put out in the urine. So that's why this blue light is important in order to remove that excess bilirubin. It essentially makes that bilirubin more water soluble. And then you can put it into the urine. And that's what this polar means. Polar just means it makes it more water soluble. All right. And this slide is kind of saying the same thing, but they're going through a lot more detail. So here you have the background of bilirubin. Essentially, it goes, and macrophages in the spleen and in the liver can help break down that heme into bilirubin. And then essentially in the liver, it needs to be conjugated with something in order to be put into the bile. So you got to conjugate it in the liver to be put into the bile. If that doesn't happen, and then once it's in the bile, you can put it in the intestine, it's going to be excreted in the feces. However, if you don't have enzymes that can conjugate it in the liver, then essentially you need to find a way to make it water soluble. Make that bilirubin water soluble so you can excrete it via the kidneys. That's essentially what this is saying. But they're using a lot of terminology, a lot of terms you don't really need to learn for the purposes of this course. And what is this about? I'm sure I put this on here for a reason. So again, this is really just about saying a neonatal, so essentially if an infant is born with jaundice, essentially can't get rid of it, then that light will help conjugate it, not conjugate it, but make it more water soluble. And this is showing how long it might take in order to remove that bilirubin from an infant. So I guess you can ignore this slide. I might have had it in there last term for a good reason, but I don't know what that was.
So does the blue light only help infants, or like in the case of like advanced like I don't know, like alcohol abuse? I know like cirrhosis of the liver.
Yeah, when you have severe liver damage, cirrhosis, which is scarring of the liver, and the liver's not working properly, then that can lead to jaundice. But at that point, you're more concerned about the liver problem than the jaundice. And so I'm sure they treat the jaundice somehow. I don't know if they use blue light on adults. I've never seen that. So, because the bigger problem with an adult with jaundice is liver damage, liver issues.
Gotcha.
With an infant, they're just not making the enzyme yet. Okay. So it's easily solvable. But with an adult that has jaundice, it's actually a really good point. It's much more severe because, an infant, it indicates they're just not making the enzyme yet. But an adult, if an adult has jaundice, there's alcoholism, fatty liver, cirrhosis of the liver, obstruction of the bile. There's bigger problems that you need to worry about when it comes to an adult with jaundice. And so we're less worried about getting rid of bilirubin and more worried about whatever that problem is that's causing the buildup. Yeah, that's actually a good point I should point out. All right, so that was heme and porphyrins. That's like half the chapter. There's a lot of stuff in that chapter. Now we're just going to really just briefly talk about the other things that we already talked about. Well, most of it. Tyrosine. What do we need tyrosine to make? Catecholamines. What are catecholamines? Dopamine. What's the other two? epinephrine, norepinephrine, right? And that's here. Dopamine, epinephrine, norepinephrine, we make them from tyrosine. All right, good. Moving on, all right? Histidine. What do we need? What are we going to make from histidine? Histamine. All right, you guys good? Okay, moving on. What do we make from, oh, what is this? Oh, tryptophan. So what can we make from tryptophan? A couple things. So we can make, darn it. I'm sure I fixed this in the video lecture too, but then when I came back to do this, I didn't. So melatonin, what makes melanin? Tyrosine. Don't confuse them like I just did, and apparently didn't change it when I fixed this slide. So I need to fix this slide again, but melatonin and serotonin, that's tryptophan. My bad. And melatonin, you should know what it does. It's circadian rhythm. So sleep, mood, light, dark rhythms, all of that's going to be related to melatonin. Appetite actually too, but so you already know that. Creatine. Okay, so this is one we haven't talked about. Creatine. Oh, maybe we did. It's a really important molecule for generating ATP fast. We need creatine. It can donate a phosphate group to ADP. And when we add a phosphate group to ADP, what do we make? ATP. So that's why creatine phosphate is a really important fuel source. If you're sprinting the first 10 seconds, you're only using creatine phosphate. So it's the very first fuel source that your body can tap into. When that runs dry, you can do glycolysis. When that runs dry, you can do the electron transport chain and the Krebs cycle and make ATP that way. The creatine phosphate is the first way we make ATP really, really fast. But we only make one per creatine phosphate. So you can only use one creatine phosphate to make one ATP. So you're not making a lot. That's why it runs out really fast. So within 10 seconds, you'll have diminished your creatine phosphate. And so what do we make it from? We make it from glycine, arginine, and methionine. So these three amino acids in order to make creatine, that really important ATP source. And the last one you need to know is glutathione. Glutathione is not just cysteine, we also need glutamate and glycine. So these three amino acids, again, very important antioxidant activity in cells, endogenously made, helps remove free radicals. We'll talk more about antioxidants next week with vitamins and minerals, well, vitamins. But glutathione is the last one that you need to know. So creatine, glycine, arginine, and methionine. Glutathione is glutamate, cysteine, and glycine. And there's your very, very few review slides for this one. All right, any questions? Let's go home, let's get some sleep, let's relax.
Not quite yet.
Oh, you guys have class. I'm gonna go home, I'm gonna get some sleep. Yes, I might do that, but yeah, sorry guys. Do we want to still talk about this?